In the Center for Evolutionary Hologenomics, we have a mission: to retell the story of life. We study organisms and their microbiome as a single unit, a so-called holobiont. Through understanding of the interactions between microbes and their hosts, we aim to crack some of life's biggest mysteries. Live animal studies have already given us some important answers, but they're often tied by technical and ethical limitations. To overcome these challenges, we need to employ novel methods and take advantage of innovative technologies.
It's extremely exciting to be part of this research that was not even possible a few years ago, and being able to explore a totally new approach for study the evolution of life.
An organoid is a simplified organ grown in a lab from stem cells. This miniature stomach, in our case, shares many similarities with its full-size sibling, but is so small we can fit hundreds of them in a plastic plate.
The simplification of the biological environment enabled us to really control the conditions, and we can monitor and manipulate the interactions between the host and the microbes. Another really important thing for me is that we can do these experiments without using live or wild animals. And most of these things wouldn't even be possible to do with live animals. These tiny lumps that you see here are our miniature stomachs that will be infected with bacteria. These bacteria are only found in human stomachs, and they can cause ulcers and cancer, but they can also protect against asthma and allergies.
Organoids represent an advanced method of studying microbial interactions, but to simulate real-life conditions even better, we enclose the organoids in a chip.
Now we can also grow these organoids in sophisticated microfluidic devices, commonly known as organ-on-a-chip. These devices artificially mimic the natural condition of the organ. When an organoid is in such an environment, they develop biological features that better resemble the real organ, and there is a chemical environment in the chip that allows us to maintain complex microbial community for weeks, which is not possible with conventional organoids.
Both organoids and the organ-on-a-chip technology are already in use for biomedical research, but we are pioneering the use in animal evolutionary studies. Among other things, we can now test effects of microbial metabolites across several different animals, entirely in a lab. The impact of host microbe interactions have been long overlooked, but to understand life, we have to study it holistically and on all of its levels. That way, we can better understand how life really works.